All right, so blood vessels. Oh God. Computer, what did you do? No, this works. All right, so we have three types of blood vessels, right? Wait, what are you doing? Okay, hold on. You and I need to have a conversation. That's still being shared. This is what happens when you try and do things at all out of order. Your computer yells at you. It doesn't do what you want it to do. Okay, that's better. All right. So we have three major types of blood vessels. Remember that we are using these blood vessels to route blood to the body's tissues, okay? Which means we need them to go everywhere. However, we also have very different environments in all of these places, okay? So we're gonna use three different types of blood vessels to get that blood to the tissues and get blood out of the tissues, as well as providing a site for exchange. So these are arteries, veins, and capillaries. They have them listed up here in order of from the heart. So we're going to go from arteries into capillaries and into veins. Now, that does not mean that we go directly from a massive artery like the aorta directly into a capillary. That would destroy a capillary. So we're going to have smaller vessels on our ways down before we get to the actual capillary beds themselves. So arteries carry blood from the heart to the capillaries. So arteries carry blood which direction? From the heart. Does it say that arteries carry oxygenated blood? No. Does the word oxygen appear on this chart anywhere or on this list? No. So does oxygenation matter for vessels? No. Okay. Just because a vessel is red on a model does not mean it is an artery. Okay. That's where a lot of people get confused somehow. Arteries carry blood away from the heart. What? They're not, because if you look at the red ones on the back of the heart, what are the red vessels on the back and sides of those heart models? They go into the left ventricle. They are carrying blood to the heart, right? That makes those the pulmonary arteries or veins that carry the blood into the left ventricle, or sorry, left atrium rather. Veins. Those are the pulmonary veins. They are red, are they not? What type of blood are they carrying? Oxygenated blood. Why? Because it just came from the lungs where it got oxygenated. So now we have to take it to the body, right? So don't get that part confused, because we have two sets of circulation here. We have pulmonary circulation and we have systemic circulation. However, they both have arteries, capillaries, and veins in that order. Okay, it's just the color of those vessels is going to be different because the oxygenation state of those two places is different. Okay, so arteries convey blood from the heart to the capillaries. We carry blood to the capillaries because that is the functional part of the bloodstream or of the blood vessels. That is where exchange occurs. That is where gas exchange is going to occur. That is where nutrient exchange is going to occur. That is where hormones will leave the blood vessels. That is the functional part of the blood vessels. All of the other stuff is highways. Capillaries are drive-throughs. This is where you hand off one thing and you receive something else in exchange. Everything else is highway. But just like if you wanted to go to a drive-through off of I-20, you have to pull off onto a service road, right? So you pull off onto a smaller road and then probably onto a smaller road before you can finally go through your drive-through, right? The same thing is going to be true of your blood vessels. You're going to go from big, massive roads to smaller roads, to smaller roads, to smaller roads, and then finally you can do the drive-through where you're going to do your little exchange. You're going to get your stuff and you're going to give them something in exchange, okay? So capillaries then are going to be very, very small, very porous blood vessels. When I say very small, I'm talking just a little bit larger than a red blood cell. Okay. They have to be big enough to allow white blood cells to pass through them, the biggest white blood cells, but that's it. That's as big as they are. That's also very important because remember, what do red blood cells look like when they pass through capillaries? What do those red blood cells do when they pass through? Do they just pass through like one at a time, just in like a constant flow? Or what do the red blood cells do when they get to a capillary? They stack up, just like cars in a drive-through or cars going through a toll booth. They line up one after the other. So they'll just stack up like this, which makes them really, really efficient, okay? But like I said, capillaries is where we're going to have exchange of substances. Because we have exchange of substances, we have to be porous here. We have little tiny holes, or really just we have holes of varying sizes in the sides of the capillaries to allow us to exchange things. Stuff like oxygen and CO2, those can pass through without holes whatsoever. Some larger things will need bigger holes, and we'll talk about that when we get there. But then we also have veins. Veins carry blood from the capillaries to the heart. Now, you've all been through a toll booth driving, right? So cars are going really, really fast until they get to a toll booth, right? So you tend to have a fairly constant flow of cars moving into a toll booth. Imagine like you're going to DFW or something like that. What happens on the other side of the toll booth? Is there a constant flow of cars leaving it? Is it though? You tend to get kind of gappy distributions from there. Like, cause like this car over here will leave and the one 4 lanes over will leave, but it's not like 12 go up, they all leave at the same time, right? That doesn't really happen. It's, they all kind of line up right there. And then as different gates go off, different cars will leave. So you have this big stretch of road, which honestly won't have that many cars passing through it at any one given time, which means you have a much lower pressure on the other side of your toll booth, okay? So you're going to have a much lower pressure in your veins than you do the arteries that lead up to the capillaries. Once you've gone through the capillaries, you're going to lose all of that force, okay? Now, to get us here, we can look at the walls of the blood vessels. Let me blow this up just a little bit more so we can see it a little better. How centered is that? Not very, but it's fine. All right, so here we have our blood vessels. So we have an artery on your left and we have a vein on your right, and we have a capillary in the middle. First, let's go ahead and look at the functional part. Let's look at the capillary. Notice we have one layer of tissue. We have a layer of simple squamous epithelium. This is our endothelium. That is it. All this stuff right here, this kind of purple, this is just the basement membrane, which remember is some extracellular protein that kind of holds things together. That's it. Really, really thin, really, really thin, so that diffusion can happen now. The thicker it is, the longer it takes for diffusion to occur. Now, let's go ahead and look at our artery, conveniently coded in blue, and our vein conveniently coded in red, and our vein conveniently coded in blue. However, keep in mind, these are systemic arteries and veins if they look like this, okay? Now, what we want to look at with our arteries and veins is that we actually have the same 3 layers of tissue. We're going to call these the tunics of the vascular tissue, or the vascular tunics. We have the tunica externa, media, and intima. Externa is, of course, going to be the outside. Media is going to be the middle. And then finally, the intima is going to be the innermost. Now, starting from the outside, we have very different external tunics on our veins versus our arteries. Look at this little layer right here. This layer right here is called the vasa vasorum. Okay, so this little layer right here is much, much thicker on the vein than it is in the artery. In the artery, it's incredibly thin. In the vein, it's quite a lot thicker. Okay, this is basically going to be the outer sheath of the tissue. So the outer sheath of the tissue is this vasa vasorum. Okay. Now, underneath that, we're going to have the tunica media. The tunica media is going to contain both this layer of muscle and this layer of connective tissue. So we're going to have a layer of muscle and a layer of connective tissue. The innermost layer will have a layer of muscle and a layer of connective tissue. Okay. So this layer of muscle and this layer of connective tissue, are these the same size between the arteries and the veins? No. They are not. Are they thicker in the artery or are they thicker in the vein? Artery. So this muscle contracts. What do you think is going to happen to the diameter of this blood vessel when this muscle contracts? Make it smaller. Right. So this one has less muscle. So if this muscle contracts, it's going to also make it smaller. Which one is going to make the diameter even more small? This one or this one? The artery. So which of these vessels do you think we use to control blood pressure? Your arteries. We can use the muscle in the walls of the arteries to control the pressure in the artery. That is because if we close this, we make the diameter smaller, which means we are increasing the pressure in that particular place. So much so that we can actually decrease the amount of flow through some of these areas by decreasing their diameter so much that it makes it hard for fluid to move through them. This way we can control the movement of blood through the system. We can shunt blood where we need it to go and shunt blood away from where we don't. So say you decided you wanted to go for a run. Well, if we're going to go for a run, okay, what needs more oxygen? My muscles or my digestive system? Your muscles. You don't digest your food very well when you're jogging or running or doing strenuous exercise. So you move blood away from your digestive organs and you push extra blood into your muscles and things like that because you're moving. Okay, so the diameter of the vessels in all of your intestinal organs would then close because we don't want that much blood moving through those areas. If we constrict flow in those places, that blood has to go somewhere else. So we can open the blood vessels to our muscles to say, hey, blood, we'll make it easier for you to move in the muscles, and we're going to make it harder for you to move into your digestive system organs. So this way we can regulate where blood is going. We do all of that with which ones, the arteries or the veins? Which one do we get to regulate where blood is going? We're going to do that with the arteries. Does that also make sense that that is how we would do that though? Because the arteries control where the blood is going, not where it's been. Is there any point in regulating the flow of blood after it's already gone to where it's done its thing? Not really. So because of that, these are just going to be open. Okay, so we have a really, really thin layer of muscle here. So that muscle and that external elastic lamina, that's part of the tunica media or the middle tunic. Wherever you see smooth muscle, it is the tunica media. Okay, now notice that I said smooth muscle. This is not something that you can control. You cannot make a conscious decision to send more blood to your muscles. Okay, that is just something that will happen based off of the needs of those tissues. Now, then we can look at the tunica intima. Okay, so this is going to be the innermost layer. Okay, so like your intimate feelings are your close feelings, right? So here we have our endothelium, which is the lining. So notice that we have endothelium, endothelium. Oh, look, it's the same endothelium all over here. What did we call the innermost layer of the heart? Anyone remember? Endocardium, which was also simple squamous epithelium. They are continuous. So the endothelium of the blood vessels is continuous with the endothelium or the endocardium of the heart. Okay, it looks the same, acts the same, does the same stuff basically. Now, underneath that, we have a what we call the subendothelial layer. So just between that, and then we have a layer of elastic tissue. Now, notice here, though, this layer of elastic tissue and this layer of elastic tissue aren't found in the veins. What does that tell us? The vein's not moving a lot. It's not having to change its shape based off of blood load. But the arteries are. Can you think of why the arteries might have to change shape because of blood load? What is happening to the arteries if they are carrying blood that the heart is moving through?
Well, they're carrying nutrients.
They're carrying nutrients and things like that, but let's think more in terms of pressures. What is your heart doing to the blood? It's pumping. Does your heart pump constantly where it's just a static flow out of your heart? Or is it in phases? It's in phases, it's in beats. So when your heart beats, bolus of blood. Heart beats again, bolus of blood. So every time your heart beats, you're going to have more blood into that system in that particular moment, which means these blood vessels have to deal with that force. They have to deal with these pulses of blood, where at one moment, there's not a whole lot. There's 80 millimeters mercury worth of pressure on those blood walls, or on those vessel walls. But at another moment, there's 120 millimeters of mercury worth of pressure on those walls. I know I did not pick those numbers randomly. Those of y'all that know any or know blood pressures, 120 over 80 is an average blood pressure. Heart is relaxed or vessels relaxed, so in between beats, that's where you have your 80 millimeters mercury. That means there's that much blood in that vessel submitting that much force on those walls. The 120 is the amount of force that your pulse is pushing against those walls. So that bolus of blood that comes out when your heart beats, or not really a true bolus, that extra blood that happens when your heart beats, that is submitting 120 millimeters of force on those walls. So you have more force whenever you're pushing the blood out. Now, you can also see that these walls get even thicker the closer you get to the heart. That's because the closer you are to the heart, the more force there is, because there's more blood going through fewer vessels. So there's more force. So you have to have even more of that elastic tissue to be able to deal with all of that force. Your aorta will have tons of elastic tissue in it because it is flexing a bunch, or has to be able to flex a bunch to be able to deal with all the force that your heart is putting on it when that massive muscle in that left ventricle pushes the blood out at really massive pressures. We don't want to have a limit to how much stretch we have, because what's going to happen to your aorta should you not have enough stretch in your elastic tissue? Or happens to your arteries if you don't have enough stretch in your elastic tissue? What would happen to the vessel wall?
Pressure makes the pipe burst.
Pressure makes the pipe bust and it breaks. This has happened to lots of people, particularly people with types of atherosclerosis. You get enough pressure on those walls and the walls don't have enough stretch because you've smoked or whatever that may be. So the vessel walls break and they shear. They call this a dissection. I happen to know someone that had an aortic dissection from here. Actually, hold on. Here, sorry, rib 7. Down to here where it splits. He almost died. He was in the hospital for like a month and a half. This is my father-in-law, actually.
What do they have to do there?
They put in stents, and made him relax and do all kinds of fun stuff. And it also led to other issues downstream because blood flow was all wonky and weird and pressures were wonky and weird. And it led to some issues with the vasculature in his legs and all kinds of fun stuff. His legs still look more like an elephant's legs than they do a human's because of all the fluid and stuff that they're holding because the vessels are all fucked. Messed up. So that is what can happen if the vessels do not have enough stretch. It is because you are pushing with that much force. Okay. Now, after all of that has happened, so we've pushed the blood through this. So we have the tunica intima, which has the endothelium, which is going to basically be the one that's touching the blood and the blood plasma. We have the subendothelial layer, so we have a thin layer of connective tissue under that. And then we have the elastic lamina, which is basically our stretchy layer. The elastic lamina are only going to be found in the walls of the artery. That's because our pressures are relatively equalized in all of our veins. After we have gone through the capillary bed, so after you have gone through the toll booth at DFW, you tend to run out of your, like you are driving now and you're no longer in a traffic jam. You're in a traffic jam basically until you get through that gate. The moment you get through the gate, all of a sudden it's like the seas have parted and everything is fine. Everybody's going 60 miles an hour and you have no problems. Okay. The same thing is happening in the veins. Once we've gone through the capillaries, we've already gone through where we're backed up. pressures and things like that tend to equalize through all of our veins. We don't really have much force. Okay. Now, one thing that our veins have that our arteries don't have are going to be these little cups, these little valves. Okay. What these are going to do is these are basically going to prevent backflow of blood through the veins whenever we don't have active flow. So in between heartbeats, our blood can kind of settle in our veins, especially if we are standing up. And gravity is kind of now trying to pull the blood back down to our feet. We want to make sure that blood continues only moving forward. So whenever your heart beats, more blood comes through that capillary and more fluid coming through, and that extra fluid is going to help push that blood up through the vein. However, and when your body relaxes, so in between beats, the blood will kind of settle a little bit, and we want to keep it from flowing back down. So we're going to do that using the valves. We don't have valves in the arteries because there is a bunch of pressure. So there's constantly some amount of fluid flow through the arteries. There's more right after a heartbeat, but there's always some amount of flow through them. Okay. All right, so let's go ahead and look at them on a microscope, okay, or under a microscope. So here we have our artery, here we have our vein. Relatively easy to tell them apart, because which one has thicker walls? The artery. Which one has a bigger lumen? The vein. Which one has a constant level of blood in it? The artery. Notice that the artery here is full. The vein is not. Okay, so there's always going to be some amount of pressure in the arteries. Okay, now if we were looking for the differences between the different layers of the lamina, it's a lot easier for me to see on my screen than it is up there on the projection. But you can actually see that this little layer right here looks more dense. It's kind of dark. Imagine like the rind on an orange or something like that. So we have this kind of dense dark layer here. Maybe not an orange, hold on. Maybe like an avocado, where there's like, you look inside of an avocado close to the skin, there's that green layer that stays dark green kind of on the inside, and then it goes into pale green. This is that layer of dark green. Okay, so we have a little bit darker, denser layer right here on the outside. So this is our tunica externa. Tunica media is almost all smooth muscle here. If we zoomed in even more, you could see the layers of the lamina, like those elastic lamina, but I'm not worried about it here. Okay, we don't actually have any capillaries here on this particular screen that we can see. Like maybe one of these guys is, but it's too hard to tell. So another thing to look at with our arteries and versus veins is the arteries, because they're going to be constantly under pressure. These are always or almost always going to have a circular lumen. Okay. There's enough tension in these muscles, and there's also enough tension from the blood that's kind of pushing through them that they're going to make themselves a circle. Okay. Just like if you ever, so say you pick up a bubble wand, and you pick up a bubble wand that is like a triangle or a star in shape, and you blow a bubble out of that bubble wand, what shape will your bubble be? It's going to be a circle. Always, every single time. You cannot make a bubble that is not a circle. Physically not possible. Because the air that you blow into the soap to make the bubble will equalize its pressure into a circle. That's because it's the most stable form. Okay. The constant pressure moving through the arteries is going to force them into a circle, because that is its most stable form. Okay. So because of that, we're going to get these nice little circles on the insides of, or the lumens of the arteries are usually going to be these nice little circles. Whenever they're sliced and put on a microscope slide, that may change them a little bit. Like the previous slide, they weren't quite perfect, but they're going to be far more circular than veins. And you and I, they are almost all pretty much perfect circles unless muscles are contorting them in some way. Veins, we don't have much pressure here, because we don't have much pressure and we don't have constant pressure on the walls of the veins. The walls do not have to be circular. The lumens do not have to be circular. They can be much more flat. So veins will oftentimes be a little bit flat. Once again, our veins are also going to have valves where your arteries will not. Now, something else to keep in mind here is as you move down in size of your arteries, so say you go from, say, your aorta into some arteries moving through the muscles, they're going to be subject to slightly different forces. We're slowing things down, just like you tend to drive faster on big highways than you do on two smaller highways or smaller roads. As we move further away from the heart, we are going to slow down as we are moving into those smaller arteries. So we're going to have a little bit less stretch. We're going to have a little bit less muscle. There will still be muscle here because it's important that we regulate it that way, but we don't have to have as much elastic tissue in the kind of peripheral arteries and things, especially not into the arterioles, okay? So as we move away, our lumens get smaller because we're moving less blood through them, just like we don't need a big 15-lane highway if we're only going to be using a quarter of the cars that were on the big road, right? So we will have a decrease in the lumen number. We're also going to have a decrease in those elastic fibers because once again, we're dealing with less pressure. However, we will have an increase in the amount of smooth muscle. That's because this is where we are going to regulate a lot of the pressure that we're moving through. This is where we're going to regulate a lot of the blood flow, is in some of these smaller arteries. This is where we can say, okay, you don't go to this muscle, you go to this muscle, or you don't go to this part of the muscle, you go to this other part of the muscle. And that's by kind of decreasing the diameters in some of these smaller arteries, what we might call muscular arteries. Muscular because they actually have a larger amount of muscle, okay? So you'll need to kind of read some of these, and you can look at all of these. These are all different types of arteries here. Notice we have really thick walls here. We're getting a little bit less thick walls into this arterial. We still have some smooth muscle so that we can still control it, but it's a little bit less than it was in the muscular or elastic arteries. In the elastic arteries, there are fewer distinct layers of elastic tissue like we have over here. Instead, we have elastic basically just spread through all of the musculature. Now, like I said before, we do have different types of capillaries, and where the types of capillaries that we're going to have is going to depend on what we need the capillary to do. If we are just in some random tissue or in some random muscle or bit of skin or something like that, we're going to use these what we call continuous capillaries. That's to say that basically everywhere within the capillary is going to basically behave the same way. These are going to have what we call penocytic vesicles. These are basically ways for the cells to drink or basically move nutrients from the tissues and into the blood, or out of the blood and into the tissues, small kind of fluid-filled things. Very small clefts in between the cells so that we can kind of move a little bit larger stuff through them, but we're mostly not worried about dealing with large molecules moving out of these things, okay? So large particles like big proteins and cells and things like that cannot move through these continuous arteries or capillaries because they are continuous. We don't see a hole big enough to let a red blood cell out of this, which means the red blood cells that pass through here are exchanging oxygen. That's it. Plasma, all of those kinds of things. We're exchanging nutrients, but we're not necessarily filtering anything out of this. This is not where we get rid of bad red blood cells. Does that make sense? Now, if, however, we move into the kidneys or into the intestines where we need to be moving slightly larger molecules, okay? Here's a question. Should blood ever be found in your urine? No, that is bad. If blood is ever found in your urine, that would basically be like you finding coffee grounds within your filtered coffee. You should not find a grind, like a piece of coffee in your coffee cup out of like a drip coffee filter. If so, that means there has been a tear in the filter. If you find a red blood cell in your urine, that means there has been a tear in your filter, which is in your kidneys, okay? However, there are lots of things that we do need to move out of our kidneys, things that are larger than just something like glucose. So we may need to get rid of proteins or move proteins from one side or from the blood plasma out or from out of the kidneys and in. Or also, this is especially important for larger nutrients. So say we're taking in proteins in our intestines. So we're eating and we're trying to take in all these nutrients. We need a way of getting those into the blood, right? So that means we have to have larger openings in the blood. So we're going to have these fenestrated capillaries. So they're called fenestrated because we have these little holes. These little holes we're going to call fenestrations. This is where we get larger nutrients into or out of the blood. So this is going to be particularly found in your intestines and in your kidney capillaries. So movement of smaller plasma proteins, but not really large ones. Also, not complete cells. However, do we ever have circumstances where we might need to get rid of complete cells? So say you have a broken down red blood cell. How do we get rid of it? Do we know, does anyone know kind of where we get rid of those things? Or how do we get new red blood cells into our blood? Where are red blood cells made? Or where are blood cells in general made? They're not made in the blood, right? So where do we make them? Anybody? Where do we make blood cells? Say you know someone with leukemia. They are likely on a transplant list for what?
It's in the spleen or the bone
Bone
marrow,
marrow.
isn't it?
They're made in the bone marrow, which means if we make them in the bone marrow, we have to have a way of getting them into the blood, right? So that means we have to have a portion of capillary in bone marrow where the cells are made with holes large enough to get them into the bloodstream. Does that make sense? Seems reasonable. Okay. So what we're going to see here is we're going to see large openings or sinusoids in our places where we make the cells. So in our bone marrow, that's because we need to get cells into the bloodstream. So that basement membrane is actually fragmented in these sinusoids. That's to make sure that once again, we have openings large enough to get complete cells from out of the blood into the blood, okay? Now, this is also important in a place like, say, your liver, which is where you get rid of old broken down red blood cells, that we also will have something like this in our liver, so that we're in our spleen, so that we can get rid of these cells as they are going bad as well. Does that make sense? So if you need to be able to replace these cells, whether it's red blood cells or white cells, we have to have openings that are large enough to allow us to do that, okay? So we're going to use these sinusoids for that. Now, also importantly here with our capillaries is that we also will have layers of smooth muscle on either end or on the arterial side of the capillaries, as well as actually on the arterial side of the capillary. That's because we can use this smooth muscle to constrict flow through the capillary, so that we don't always have to have active like we're not always pushing blood through every single capillary that we have in all the places. That's because we have different requirements at all times. Your tissues have different oxygen requirements based on whatever it is you're doing. Your tissues are also producing more waste or less waste at different times. So they may not always need a way of getting rid of that waste, those waste products and things like that. So we don't always have to have capillaries open. However, we can open capillaries when we need to by relaxing these smooth muscle cells to allow those blood or allow that blood to go to those places. So like before, I said like you started running, right? Well, when you start running, that means we need to increase the amount of blood flow to those muscles. Okay. What else is, what is another place where we're going to see increased blood flow when you start running or you've been running for a few minutes? What else starts to happen? What color does your face turn? Turns red, you flush. Why are you flushing? What's the point of that? Are you only flushing just because you have increased blood flow everywhere? No. To cool off. Exactly. So we're going to open the smooth muscles around the capillaries of your face and your skin so that we can expose, or so we can send more blood through those places so that we can cool it off. So that we can use the outside environment as a heat sink, so we can use that to dissipate all of the heat that we're now making in our muscles. Because it's relatively cold outside, if you walked around outside or even just like sat still outside, like on a bench when it's cold, what happens? What do you tend to do? You shiver because you're using your muscles to generate heat. However, if you use all of your muscles to generate heat, you'll eventually get to the point where you're generating more heat than is necessary to keep you warm. So you need to dissipate some of that heat. So we're going to use our skin to dissipate the heat, which means we open up the capillary beds in our skin, so your skin will flush. So your skin will get a little bit more red because we're moving blood to the surface so that we can cool it off. Does that make sense? So we can control where that blood is going using these smooth muscle cells. If we don't want blood to go out there because, say, we don't want to lose heat, like when we're shivering, these smooth muscles constrict. do you tend to get pale or red when it's cold on non-exposed areas? So where I'm not talking about like getting wind whipped or anything like that. You tend to be pale. That's because your body wants to hold on to that heat. Like if you look at someone's face and they are red, that likely does not mean that they are hypothermic. What color does someone turn when they are hypothermic? They turn blue, right? That means we don't have a whole lot of blood at the surface. That's because their body tissues are holding on to blood, so all these arteries are, or the arterial end of your capillaries in your skin would be closed off to prevent blood from going to those places to make you colder. Does that make sense? So we control that once again using smooth muscle. And we can also use this to kind of restrict blood through the peripheral portions of the capillary bed too, to make it basically just shunt over into the veins and not do much exchange. Now, veins are going to have the same kinds of things. We're going to go from arteries to arterioles to capillaries. Veins, we're going to go from the capillary to a venule and then finally into a vein. Now, one thing that veins do act as are blood reservoirs. This means they're going to hold on to more of the blood. Most of your blood at any one given point in time is going to be found in your veins. Okay, 55% of your 5 liters of blood will be found in the veins. Only 10 is going to be found in the arteries at any one given point in time. Look at this. Your heart has 12% of the blood at rest. So when your heart is filling, or when your heart is filled, basically, it will have 12% of your overall blood volume. That is more than what you will find just in your arteries. That's kind of crazy. And another 18% is going to be found in your pulmonary circulation. So your veins are basically where most of your blood volume is going to be in any one point in time. All right. Now, something else to keep in mind, like anatomy-wise, if you're wondering what I'm doing by skipping slides, like I said, we're mostly concerned with kind of anatomy stuff in here, and y'all can read the physiology stuff for lecture, okay? We don't always have these perfect little artery to arterial to capillary to venule to vein things. Instead, sometimes we're going to have what we're going to call an anastomose. An anastomose is basically going to be a network of blood vessels all leading to a capillary or on the other side of a capillary. So like, look at the back of your hand. So if you look at the back of your hand, if you can see your blood vessels anyway, if you look at the back of your hand, you'll actually see a network of veins on the back of your hand where they are all interconnected, okay? So like if you constrict a little bit of blood flow, so some blood kind of pools in there, you can actually see that they're all kind of intermeshed together. There's no one input or one output for those veins. They're all kind of a mesh, okay, or a network. So that is an anastomose. Anytime you see multiple things all connected together that aren't necessarily coming from one place or going to one place, that is going to be an anastomose. Now, in this case, the one on the back of your hand would be a venous anastomose, but we also have arterial anastomosis in there as well, okay? So we're just naming it based off of what the primary or what the vessels are in that particular spot. Now, importantly, we also have a portal system in some places. Now, these portal systems are going to basically be where we're going to have dueling capillary beds, where we're going to have a capillary bed in one place, which is where we're going to do all of our nutrient exchange and all of that kind of stuff. But then where we're going to have a capillary bed on the other side, which is another place that we can do more exchange. So if we need to get blood cells out of the vessels and things like that, we're not going to do that in a capillary where we're also exchanging nutrients and we're exchanging oxygen. That would honestly be kind of silly because you might lose some of the red blood cells that have oxygen you might want and stuff like that. So we tend to do all that kind of stuff downstream of our nutrient exchange. So this is going to be particularly important in our liver, where we're going to have our hepatic portal system so that we can clean out the blood on the other side of where we've actually already done our nutrient exchange. So we'll do the nutrient exchange at various organs in our abdominal cavity and then have it go through the hepatic portal system so we can clean out any bad blood cells. Okay, so the next thing that we're going to be needing to worry about then is how we look at blood pressure. Now, blood pressure is basically just going to be the pressure that is exerted on the walls from a pulse. So a pulse is basically just the rhythmic throbbing of the arterial wall from the heartbeat, okay? The rate that you see these increases in pressure, so those pulses, that throbbing of the wall, that is basically what you would use to time like how often the heart is beating. But keep in mind that your heart kind of beats in two ways. We have a beat in the atriums, and then we have a portion of a beat in the ventricles. What you're feeling as the pulse pressure is actually the beats of just the ventricles, okay? But if you were to listen to someone's heart sounds, you hear like lub dub, where you're actually hearing those valves shut, okay? That is a little bit different than the actual pressures, okay? What you're feeling as a pulse of that throbbing you feel is basically going to be the systolic pressure or the stretch that is placed on the blood vessel by the heart beating. So by the ventricles contracting and pushing the blood out, they're exerting a certain amount of force on the fluid and on the walls of the arteries that are holding the fluid, okay? So whenever you're taking someone's blood pressure, what you're really doing is you're going to take a cuff and you're going to squeeze the artery, okay? You want to exert so much force that the heart cannot push blood through that pressure, okay? So that's why whenever you're pumping up the cuff or whenever the cuff is inflating, it inflates well above an average blood pressure start, okay? That's because we want to shut off flow through that particular vessel. And we're usually picking a superficial one so that we can actually shut it off. Okay, if it's deep, we're not going to be able to put enough pressure on to shut off blood flow through it. That's why whenever you're having your blood pressure taken, there is still blood moving down here. It's just from deep vessels instead of the median cubital artery that we're going to use. Okay, so you take the cuff and you cut off blood flow through that particular vessel. Now, when you slowly release air from it, what you're doing is you're slowly decreasing the amount of pressure that is placed on the blood vessel until eventually you'd hit a point where you can start to hear the pulse again. The moment you start to hear the pulse again, that basically tells you this is how much force is placed on these walls at this particular point in time, okay? That is how much force is required to overcome the vessel or overcome the pressure that you're placing on it and allow blood to move through it. That is how much force the pulse is putting on. That is your pressure that we would call the systolic pressure. That is the top number. So when you start to hear the pulse again, that is your systolic pressure because that is the maximum amount of force that that vessel or that the heart is placing on that vessel. So that is your systolic pressure. What that is though, that's basically the amount of force that the ventricles are pushing out with. That's how much force they're placing on the fluid and pushing it through the system. Now, I said before that your arteries also have a constant flow of pressure, right? So in between beats, so you have beat, beat, beat. In between, there is still pressure on those walls. The pressure that's on those walls is going to be what we're going to call our diastolic pressure, okay? Now, your diastolic pressure, like I said, is that constant pressure that's placed on the arteries as those arteries are kind of like recoiling, going back to normal. You're going to lose the sound when you are trying to listen to someone in this case. That's because that is basically just how much pressure there is on it when the fluid is moving through it. When you stop hearing sounds, that is going to be your bottom number of your pressure. So when you start hearing sounds, that is basically saying, hey, this is how much force is being placed on the arteries that you can now hear. When that sound goes away, that is your diastolic pressure. And that's basically saying, okay, this is how much recoil we have in the vessels, okay? And how much pressure there is in those vessels that kind of rest, essentially. So during ventricle relaxation. So we have ventricles contracting, ventricles relaxing, ventricles contracting, ventricles relaxed. So we're going to go between those things. The difference between those two is what we're going to call a pulse pressure. That is basically to say, hey, this is how much pressure there is in between these two numbers. That's what we're going to call a pulse pressure. And that's what you actually feel as a pulse, okay? This is typically in kind of an average person in an average time going to be around 40 millimeters of mercury. So the first little exercise we're going to do today is actually going to be to take some of these blood pressures. So to do that, we have cuffs and we have stethoscopes back here. We have some automated ones. Don't use those. They're not fun. Okay. So we have blood pressure cuffs and we've got some stethoscopes. Now, the fun thing is that some of these stethoscopes will actually have two sets of headsets on. So two people can actually listen. So the person that is acting as the patient or the subject can listen while the other person is taking it, and y'all can both look at the little chart right here. So what we're going to do is we're going to take a cuff, and usually want the person's arm not extended, but not fully bent either, because that'll kind of constrict flow. You want it basically just relaxed. You want it sitting on a table, not hanging down. You hang it down, pressure will change. Okay, we're going to wrap this around. Most of y'all have probably already done this to someone at some point, so you're going to know this even better than I will. You wrap this around, and we're going to hold the reading point, or we want to basically hold pressure up here, and you're going to hold the face of the stethoscope on our median cubital artery. So cubital meaning elbow, right? We're basically going to hold this slightly to the left, so slightly medial of midline on the arm. Not perfectly in the middle, just slight bit left, because that tends to be where that median cubital artery is going to be. That's sort of the base of the cuff. Then you inflate the cuff until you shut off blood flow. Okay, so you're going to inflate this a lot. If they say it hurts, that means you're probably going hard enough. Okay, but when you listen to the stethoscope, you can still feel or still hear a heartbeat. You keep going. You keep going until that number, or until you can't hear blood flowing through it at all. And it is going to be a little painful. I didn't have this tight, so that's not very attractive. Okay. Then once that happens, you're going to turn this little dial. Oh, that's nice. Okay. You're going to turn this little dial to slowly allow the air to leak out, and then you're listening for when you can hear the pulse again. When you hear the pulse, that is your top number. You're going to continue slowly releasing air until you lose your ability to hear it, because that means there's no longer enough pressure being placed against the stethoscope for you to hear. So that sound will go away. When that sound goes away, that means you have reached your bottom number, which is your diastolic. That is your blood pressure. And then you can use that to calculate your pulse rate. And then we're going to do some other calculations with that after you are done with that. So we can look at mean flow rates and stuff like that. So save those numbers. You're usually best to take a couple of numbers, but then we're going to do something fun in here as well. Namely that I want you to do it twice on whoever's doing it. Honestly, everybody should probably take their blood pressure twice. Then we're going to have somebody go outside and run a couple laps around the hallway, and then come back in and we're going to check it again. And I'll show you that number is about to change. Not run, just walking, or slow walking will do it, and even like a brisk walking, you can actually see where the numbers even change. If you really want to be mean, you make someone literally go run some stairs and come back, or have somebody like rip up on the table. There's all kinds of fun stuff you can do. Or someone could lay down on the table and just like close your eyes and listen to classical music and see the number fall. Okay, so I'll give you all 30 minutes or so to mess with this kind of stuff. Everybody needs to check their blood pressure with these twice. Once again, you have two subject pieces here. Use an alcohol swab to keep these things clean, that kind of deal. Everybody good? See what we're doing? All right, let's have some fun. No, y'all cannot check mine. I don't remember if that's my blood pressure medication this morning, and I'm a little scared.
It's not gonna work like that just one day.
Huh?
It won't go up just one day. That much.
Well, I didn't take it. So like I took it yesterday, but mine's basically a 24-hour deal.
And you're good then. You can still get a good reading. It's kind of like blood pressure medicine, kind of works a little bit like birth control. It gets into your system.
No, that's not how that works at all. Like after the full day has gone off, it's like the opposite will actually be true, because
Continually take it. Just because you come to the doctor's office and you didn't take it that long, it's not.
yes, but it is actually important that you try and take
Yes.
them at not even just this every day, but the same time every day, so that you can keep the cycles all the same. Because what will happen with at least my pressure medication in particular is you'll actually see a rebound effect with mine, where if you don't take it for even a day, like when the last of that drug is gone, you'll actually see a larger response in the vessel. So you'll actually have an even bigger increase in pressures. So you would look at it and you would see like a spike where I might be like 160 over 130 on a day that I didn't take it. I say this because I have also checked it because I didn't believe my doctor when she told me that. And then, yeah, no, she was right. So yes, I did purposely not take my blood pressure medication one day just to look at that. And yeah, he was right. Because I'm on it because my resting blood pressure was in like 140 over 110.
Mine the other day, I told my doctor that I was,
Yeah. Why? Because I have a test today. So you want to snug these cuffs down at least somewhat snug before you start squeezing. Otherwise, you'll just be hitting that button all day.
Very small.
like the little leg.
Yeah.
Takes like 3 whole pumps to get it to 200.
And then they want a shot and you're like, you're gonna go straight through her arm.
Those are the ones you don't have to do CPR on because they're great. I told you about that one night my wife broke 3 sets of ribs. Because of little old ladies.
Well, I gotta get it off.
Sometimes you can also just see the needle jump.
Mr. Mosley.
God, your blood pressure is literal perfect. It was like 120 over 70. Jesus.
Mr. Mosley.
What's your blood pressure? Textbook.
I just went to the doctor yesterday. It was 124 over 78.
And your doctor's like, well done. Everything looks good.
I have to go for migraines. Yeah, I changed my medicine. And it makes me so.
Yeah, the fun bit with migraines too is that there's lots of ways that those can hurt.
Yeah, they're making me start an injection for me.
What's all 58? We need to write that down.
Where you feel like that whole divot. Just slide over the left.
Okay, do you understand this?
So it's
This
basically
is $3. $3.
slightly-
Okay, I was going to listen with you.
So you should be able to, when you get some pressure in it, you should. Normally, right?
I'm gonna not put mine on until you get her set up. It's this way, it's all out, right? Okay, go ahead and turn it to the right. Go to the right, got it. Okay, now it's all the way down. Once you go ahead, so you get used to loosening it, go ahead and pump. You're gonna have to use one hand, and then
So the first one says you're
so
going.
what you're gonna do is you're gonna have one hand. so
Another trick is
You'll have to
honestly.
focus on the part here, and you're gonna.
Could you hear it?
up? No, practice kind of letting it go slow.
Oh, did you not have those? Oh, I need something. So if you want to kind of practice just hearing it, you can actually just kind of put it on this. You'll be able to hear it. Without much pressure. Or you can just listen to their heart sound. Or you can hear the sound. And then you. Or you can also just kind of pump it up to like here or so where you can pressure deep and just listen. Get it just what it sounds like and then you.
Yeah, that was good, okay. Now we're ready for the little bit. Hold this right hand.
Let's just. But I'm talking about the.
Is it all the way out? Shut it off. And you're gonna hold that. That's tighten it all the way.
Because the pressure is changing.
You're gonna wanna hold it, don't hold it with your thumb. They say to hold it. Squeeze like one day, yeah. I'll at least do it to like an hour. That will be really slow. And see, that's why I said practice. Okay, you could just hear, all I could hear is like the bottom two right there. Here, help me out.
Does that make sense? Could you hear it the whole distance? Or like, could you hear it like when you were within the blood pressure?
Just try to listen.
How we doing?
Good.
You did that? 88 seconds. Good, what'd you get? Where did you hear the first beat? This time it was 110. Yeah, we not really good. But you can hear pretty good. I'm surprised that there's two ways there.
You'll see it again.
I've probably done it 15 times today already.
Wasn't that what you did?
Actually, don't remember all that stuff.
Where, first you want to squeeze it and then squeeze it and see how that kind of goes. Tighten it, because you're gonna have to do it with one hand. And you're actually have to do it, you have to do it with one hand. And then just be able to do it. I tell you, I can. Squeeze, squeeze, squeeze. No one's in the doctor's office ain't gonna do this. All the way close, otherwise air is gonna be. Want kind of slow because.
So if we are looking, like, one common number that comes up in determining different things is what's called the mean arterial pressure. So mean tells us average, right? Arterial pressure. So the average pressure in the arteries at any one point in time. To calculate that, we can actually use this pulse pressure. So what we're going to see for mean arterial pressure is basically going to be the diastolic pressure plus the pulse pressure divided by 3. Okay, so to calculate mean arterial pressure, what you would do is you would take, so MAP, so mean arterial pressure is the diastolic. So that's the bottom number, right? Because we know that's the lowest amount of pressure in there at any one given point in time, right? So it's going to be the diastolic pressure plus pulse pressure over 3. So say you have someone that is our perfect 120 over 80 here. So plug that in. So the diastolic pressure is 80 plus 1/3 of our pulse pressure. So pulse pressure in this case is 40 over 3. So 40 over 3 is what? 13 and thirds. Okay. So that means it's 80 plus 13. So our MAP then in this case would be equal to 93.
Ooh.
So that is your mean arterial pressure. So we can use that once again as a different diagnostic, but we can also plug this into other formulas later on to look at bulk flow rates and things like that.
Really with this.
So here's kind of a map of blood pressure over your different blood vessels.
Usually that's when they.
So once again, like I said, we can use that for bulk flow rates, but also we can use this to tell us if we have sufficient blood flow. Okay. So if a MAP, so mean arterial pressure, is less than 60, that means we don't have good blood flow to a tissue or to a place, which can lead to problems. So here is an average or where your blood pressures or where you expect these pressures to be. Here. All right, those of y'all that can't hear, come here. Listen to mine, I promise you'll hear it.
That would be.
That's because my vessels are larger because I have more blood than y'all and I'm pushing it through with more force because, well. So you will hear it. Jesus.
I know, I love that.
No, you're not, I would die.
They looked me on blood pressure medicine and it brought it down normal. I felt so lethargic.
I don't want to be cold. Oh yeah, I can always tell on days that I haven't had my blood pressure meds, my blood pressure meds, because if something aggravates me, I can literally feel it. So if you're having problems hearing, you can
You're
come
good.
to
You're
the
gonna
side.
listen.
It'll be off. If I lay on my pillow wrong at night, I can hear my pulse.
Okay, we know. And see, another thing, you know how early.
That's not even a joke. I can literally feel my pulse.
You don't want to start getting it, see how you adjust it up at the beginning. You hear it?
Yes, funky.
130 over
What'd you
90.
get?
What?
130 over 90.
Yeah.
132. Huh?
Oh, it's because I'm talking. All right,
Mine probably works for yours.
go right around. Go walk a few laps. Go walk a few laps. Go walk a few laps.
After the gym.
And then you lay down. The people that are staying in here, like, don't lay down, just like sit back, relax, listen to like calm, quiet music, close your eyes.
He said the old people have gotta go.
Yeah, I'm Lily.
Yeah, she's probably the youngest one.
How are you? 16.
Shirins. I literally have a jacket that I still wear that's 20 years old.
I'm not gonna go there. I bartended more than she's been around.
That's a bit of an exaggeration, but I do have a jacket that's
Oh,
14.
You good?
It won't take much, just three laps or so.
hey, could you wear my watch? I could use the steps. Just hold it in your hand.
But I told you you'd be able to hear mine. It's kind of hilarious, like when you take people that like are having problems hearing, like, y'all are listening like a little tiny person, like here's mine. Mine will be so obvious the needle jumps 20 pounds.
In
They're
my office,
20.
everybody always wanted to take one for your blood draws. Everybody always wanted to draw my
Oh,
blood.
I need. I was getting IVs.
You couldn't poke your finger the other day.
Well, I don't like you. Didn't.
I'm off, like, no, just living, please.
I know.
No one has ever missed a blood draw on me. I'm not real sure it's possible.
Well, what's this? She's about killed me. She was digging up in there, that awesome one.
I have heard that that's not true. Thing.
Yeah, I didn't think it did. My dad was like.
I was told that.
They can move.
I was like,
I mean, I can.
They don't
I can just
spin.
move my ankle, I can move it back and forward.
Yeah, they don't spin, but you can make them move.
my dad said I have an artery right now.
Well, that's why I did not believe it. I can make it.
I can see it like bumping
All right, check them. The real fun one is when you take someone whose pressure is actually somewhat high, like you have, and then make them do something that's like strenuous. Like I had one person like, oh, they didn't want to walk, so they started doing like burpees and stuff. Like, okay, sure. Like a 40 millimeter spike.
So you're saying that's what it's gonna be, how you get on the treadmill, mine's gonna be like 180,
You can see massive spikes.
200.
Like you can see.
I'm gonna die.
I've also seen some, I've also seen people like athletes and stuff that are super fit. And like you take theirs even after having them do something like that, it'll only have gone up by like 10 or 20. Did it go up? Just barely. It shouldn't go up by much. Like I didn't make him run a marathon. Like it shouldn't go up by much, but it should go up. But look, that's significant. Like that's a anywhere from 10 to 14 millimeter jump, right? So that's a pretty big jump. Oh, over here, sorry. Yeah, so like that's still a 10% increase, right? But I mean, if you think about it, but 10% is a lot. Like,
Sorry, it was on. inside out.
but keep in mind that part of the reason this is also happening is because we need, like, whenever we're doing activities, we need more blood to go into our extremities to do those things. We need more blood going into our muscles. So more blood going into the muscles, which is going to mean that we're going to have more blood moving through them. So we're going to see an increased pressure. You'll also notice that someone's pulse rate increases as well. So if we increase pressure and we're increasing time under pressure, that means that we're not just getting 10% more blood into the area. If we're also pumping 10% more, that means we're getting 10% more blood from pumping faster, and we're getting 10% more from there. So we're getting another 10% bump. So we can quickly stack up a whole lot of extra blood moving to these tissues by increasing blood pressure and increasing pulse rates.
Not really. You weren't working hard enough. You needed to act like.
You probably should have walked up the stairs. Well, you can force heart rates up going upstairs. Even people that are fit, even if you just walk up 2 flights of stairs, it's enough to like cause you to get not lightheaded or anything like that, but it forces enough muscle or blood into your legs that you have to raise them. So even if you're very, very fit, you take someone that runs marathons, you make them run up 2 or 3 flights of stairs, their heart rate will increase. Just it has to. So once again, we can use this to calculate pressure, but something else to keep in mind here, just like I used that road analogy earlier. So like we're taking a bunch of pressure on, say, a highway, like pulling into DFW, there's a whole bunch of cars moving in, so you have high pressures, and then you go through the capillary and things slow down, so you're going to decrease pressures on the other sides of the capillary. So we're going to have around 20 millimeters of mercury in pressure on the venous end of a capillary, 40 on the arterial end. That is much slower or much lower, much lower than what we're dealing with in regular arterial flow. And y'all can keep messing with these things if you'd like to. It can be fun.
If you're going into nursing, get used to it.
No kidding. Now, something else to keep in mind here is that if we're looking at 20 millimeters mercury in pressure on the capillary side, that means we're only dealing with at best 20 millimeters mercury worth of pressure at all in the veins. That's not very much. That's actually not going to be enough to return the blood to the heart in a nice constant stream. So we're going to have these pulses and lags. Or we're going to have these kind of lags in pressure as we're moving through the veins. We want to make sure that we don't get backflow. To help us prevent backflow and help us keep things nice and static, we're actually going to be using our muscles to kind of help move things through. So your muscle, you're going to have a little bit of contraction of skeletal muscles on either sides of the blood vessels to help keep pressure in the vessels to make sure that the blood is constantly moving through. So once again, we have blood being pushed, blood pushing itself. We've got valves to prevent backflow, but we've also got these skeletal muscle pumps basically pushing on either side of the vessel to keep the pressures relatively even, to keep blood from pooling anywhere. So if you have muscles on either side of the vein, those muscles contract a little bit, they'll put some squeeze on the blood vessel, once again, helping push the blood through them. Because we don't want pressures to, or we don't want blood to get stuck anywhere. Now, this is primarily going to be important below the heart, okay? Because gravity is helping us bring blood to the heart in the upper body. Gravity is preventing us getting blood back into the heart in the lower body, because this blood, gravity is pulling down on it, so it can't get there. However, keep in mind we have superior vena cava where the blood's going to be pulling down here. So we're going to get help from gravity in this case to bring that blood back down. So we don't need a whole lot of muscle compression in our thoracic cavity. That's all just physiology stuff. Renin angiotensin, we'll get there later on. All right, so this is kind of what we were simulating a little bit. I know it wasn't strenuous exercise. However, during strenuous exercise, once again, we don't need blood going to our digestive organs. You're not worried about digesting your donut from this morning whenever you're running a marathon. So what we're going to see is we're going to see these numbers not just not go up, we're going to see them not, we're not just going to see them not go up, we're actually going to actively see these numbers go down, okay? That's because it is not important for us to get additional blood flow into these types of areas, so into things like your kidneys, into things like your abdominal organs, your digestive organs, whenever we are doing strenuous exercises. It is far more important that that blood is going to the muscles that are actively doing the work. So we're going to send a whole lot of blood. That's a 10 times jump for those that can't do math. Okay, so going from 1,100 milliliters a minute going to your skeletal muscles to jumping to 12,000 milliliters a minute. Okay, that's a massive jump. Why? Because they are actively being used now, not just for kind of sitting still and maintaining posture and things. Now we are actively using them to do the thing and to move you around. So we vastly increase the amount of blood flow that's going to them. We're also going to see a whole lot more blood going to your skin. Why? We talked about this before, and it's to cool us off. Okay, exposing more of that blood to the capillaries of the skin so that we can kind of use the outside environment as a heat sink, so we can dissipate some of the heat that we're generating in the muscles. Because now your muscles are generating a whole lot of heat. Like you tend to get warm, even just like walking a couple laps or something like that, even when it's cold. Like, have y'all ever noticed it, like just walking across campus or walking around when it's cold outside, you can still start to sweat a little bit? That's because your muscles are like churning through energy at that point. Because of that, they're generating a heck of a lot of heat as waste. So we try and get rid of it by increasing the amount of flow to our skin. Your heart also vastly increases in its blood needs during these types of times. Your heart's beating faster, right? And it's beating harder. So if it's beating faster and harder, it's a muscle, so it still needs extra blood going to it. So we're going to see big jumps in blood going to the muscle. We're also going to see some big jumps going into your brain, because your brain is actively telling your muscles what to do. And it's, remember, also having to coordinate all of these things, making sure they're happening at the right time. So that is actually going to put some extra stress on your brain. So you'll see it increasing its energy consumption. Now, this is saying at rest, as in you're also actively not thinking about anything. Say you picked up a chapter like this and you started reading this figure and trying to understand what this figure means, why it means this, what all these numbers mean, how they're all put together, and you're actively thinking really hard. Those kind of things will also increase the amount of blood flow that's going through your brain. You're going to need increase in energy consumption in those places. So you will actually see increased blood flow in those types of environments as well. Sitting down, doing math problems and stuff like that, you'll actually increase the amount of blood flow that's having to go through your brain to do that. Sitting in this class. Standing up here and yelling at y'all all this stuff. Now, pulmonary circulation is also a thing, okay? So we do have circulation through our hearts and lungs, or through our lungs, remember. So this is going to be dealing with lower pressures than in arterial, or than in systemic circulation. That's because we don't have to send things very far. So the right side of the heart doesn't have to push nearly as far. So it's not trying nearly as hard to do that. So these are not going to be dealing with the same amount of pressures. It's also important that we're using fairly slow blood flow here. So we don't want to like make these blood cells move through here at the speed of sound or something like that, because we want them to take their time moving through our alveoli so that we have gas exchange happening nice and fully. We want to get as much oxygen into the tissues as we can. We want to get as much CO2 out as we possibly can. So we want blood flow to be fairly slow. All right, so let's go ahead and track blood flow. This should be a little bit of a review from last time too. So we start in this chamber of the heart. What is this chamber of the heart? The right atrium, perfect. So blood drains into the right atrium, both from the superior and inferior vena cava and in from the coronary sinus, right? So then we're going to drain through this valve, which is what? Well, we're going to go into the right ventricle, but what do we call the valve? The tricuspid valve, okay? Or the right atrioventricular valve, okay? Right AV valve or atrioventricular. If you write AV, I'll count it wrong because AV could stand for audio video.
But if
Okay.
it was tricuspid, we're good.
Tricuspid's fine because it's one of those technically correct kind of things. All right, so into the right ventricle, then up through the pulmonary trunk. There's a valve here. I'm not worried about the valves. I was mostly just giving you all a hard time. All right, so what do we have here? Out of the pulmonary trunk into the pulmonary arteries. Perfect. Pulmonary arteries. Notice they're blue because we're still doing deoxygenated blood, right? Then we're going to go out here. Pulmonary arterioles, and then finally into the alveoli of the lungs. The alveoli of the lungs, we're going to have pulmonary capillaries around those alveoli. So the alveoli are these little sacs, and they've got these little pulmonary capillaries in them. Okay. Now, pulmonary capillaries, then we're going to go back through the pulmonary venules into the pulmonary veins. So the pulmonary veins drain into the left, what? Atrium, into the left ventricle, out through the big blood vessel here, which is the aorta. Very good. Good deal. So that's how we're going to do that. So that's how we're going to get oxygenated blood into the heart. Okay, so we oxygenate it in the lungs, we route it back through the venules, and then finally into the pulmonary veins, then out through the left side of the heart and out through the body. Okay, short distance, so we don't have big pressures here or anything like that. We're not having to push real hard. So we're going to have very different blood pressures found in the pulmonary circulation versus systemic circulation. All right, now there's a bunch of blood vessels. That's because we have a lot of tissues and we have a lot of places to get blood. Now, one great thing is that most of these blood vessels are going to be named for exactly where they are. If it is in your armpit region, it's going to be the axillary. If it's in the arm, brachial. If it's near your radius, it'll be the radial. Sorry, radius. Radius, it'll be the radial artery. If it's near the ulna, ulnar. If it's in the middle, it'll be called median. If it's in the cubital region and it's the one in the middle, it'll be the median cubital artery. Okay, this is where learning all of those terms and stuff that we had y'all know fairly early on are going to be very, very important. Now, coming out of the top of the heart, it's a little easier to see it on this image than it. I think there's a next one coming. But right here, we have three big vessels coming off of the aortic arch. BCS. Remember, BCS. Remember what? BCS. Brachiocephalic, cephalic, subclavian. Brachiocephalic is the first one. Second one is cephalic. The last one is the S, so is subclavian. Brachiocephalic, cephalic, and then subclavian. Okay, so BCS. Brachiocephalic tells you that this vessel is going to split into a brachial portion and a head portion. Brachio meaning arm, cephalic meaning head. So if we look right here, we're going to see this first branch is going to be the brachiocephalic because we have one branch that goes into the head and we have one branch that goes over to the right arm. The C and the S doesn't have to do that. The C and the S we're dealing with now on the left-hand side. The C goes straight to the head. The S goes into the arm. Okay. Then we have the abdominal aorta or descending aorta, finally into the abdominal aorta, and then it will split into our or in our hips, so that then we have our common iliac arteries. So the branches of the Y are going to be our common iliac arteries, and those will have a bunch of other branches. So once again, we're going to be naming things for where they are. One great thing is that most vessels are going to have, like, most of the time, if there's an artery, there will be a vein, okay? But even if there's not, where it is will also tell you what its name is going to be. So in a lot of ways, this quiz of asking y'all about the names of blood vessels and stuff like that is also in a lot of ways really just a quiz of body landmarks, okay? So don't get super stressed about it. It doesn't mean that you don't need to study these things, don't need to find them, don't need to follow their little pathways and things like that. Pay attention to the stuff that's around them, as always, okay? But if you look for a body region and you look at the stuff around it, you can usually create your own kind of diagram in your head to do this. Another good way of doing this would be to kind of basically have a drawing of an arm or a trace of an arm on a table, and then kind of lay out where you think those vessels would be and just kind of draw them for yourself and stuff like that. That can go a long way too. We do have a difference in the head of what we're going. So in the arteries, we're going to call those carotid. And the veins, we'll call those jugulars. We have internals and externals of both. Facial tells you it's going to the face. Occipital tells you it's going to the back of the skull. Auricular, auricle, that's part of the ear, right? So this one's going to be in that kind of ear region. Thyroid tells you it's going to the thyroid gland. Pharyngeal tells you it's going to the pharynx, which is your throat. Okay. Lingual. Lingual means tongue, right? Use your body regions. If you're using your body regions, this will not be hard. If you want to make this part difficult, pretend that the body region stuff doesn't exist and you want to learn all this stuff fresh, by all means do that. You will make your life a living hell if you want to do it that way. Instead, if you look at your body regions, this gets real easy, real fast. There's honestly not very many new ones that you'll need to know. Here is one of the new ones that you'll need to know right here, though. This is the cerebral arterial circle, okay? So I said before that sometimes we'll have these little networks of things that we're going to call anastomoses. Well, here's a very important anastomosis in the head of the arteries. This is oftentimes called the circle of Willis, okay? So here we have a little circle of Willis. You can see that we're at the bottom of the diencephalon here. So we're going to see these vessels kind of working their way around. As long as you can point at this and say this is the circle of Willis, I'm good. I'm not worried about necessarily you naming the ophthalmic artery or anterior cerebral artery or anything like that within the circle. But if you want to see what the breakdown of the circle is, this is a great figure for it. That's part of why I'm talking so fast right now, is because if I point at this thing as a whole, say circle of Willis, we're good. All right, so here's our veins. So we have a vein running through those transverse foramen. That's going to be our vertebral vein. We've also got our internal and external jugulars. So internal, external jugulars. So the jugular veins, these are basically going to be venous blood return from the head and through the neck, once again into the brachiocephalic. So brachiocephalic, arm and head. So kind of makes sense now that that would be the brachiocephalic vein, and then finally superior vena cava. Now, one other thing that I will point out here, though, on some specifics, we do have some dural venous sinuses that we talked about when we talked about meninges. This is once again where we're going to be kind of pulling CSF out of this, and also where we can kind of return blood back into circulation. So all of this stuff is going to drain into the internal jugular vein. But we can see a big sinus up at the top. So this would be the superior sagittal sinus, inferior sagittal sinus. We have what's called a straight sinus right here, which is basically the path that we're going to go from the internal sagittal sinus into or towards the occipital sinus. This little line right here is called the straight sinus, but we have this one cool spot right here where all of these sinus returns are going to lead to this one little area right here. This is called the confluence of sinuses. So if you can imagine that we've got a straight line here, a straight line here, and a straight line pointing back through them from the center of the brain, they all come together at a single spot. That is where your parietal lobes, or your occipital lobes would connect to your cerebellum. Okay, that area right there is going to be called the confluence of sinus. Confluence. Confluence. So a confluence is basically where things come together. Those of y'all that like your like fantasy literature, you've probably read this word before. But a confluence basically just means that things are coming together. So this is where these sinuses are all coming together, is this confluence of the sinuses right here. So we can see some more arteries here. This is basically just y'all looking at some stuff, unfortunately. We have some great pathways written down. I've also included a couple of pathway worksheets in the Blackboard section for learning this stuff. Phrenic. So if you ever see the word phrenic, this means that we are dealing with the diaphragm. Okay, so the phrenic is referring to the diaphragm. So phrenic arteries carry blood from the aorta to the diaphragm. Phrenic veins are going to carry it from the diaphragm to the vena cava. Mesenteric. So if you see mesentery, that means we're dealing with the intestines. So if y'all didn't think this class was enough of a medical terminology class, now you get more. So there's a little bit more of that there. I don't think that figures in any of the quizzes, so I wouldn't worry about it too much. Once again, we're basically just kind of naming things with where they are. Same with the arms. These are great places for you to remember particular body regions. So axillary. So here's our brachial artery, axillary, median cubital, ulnar, radial, median. This should all be fairly straightforward. Okay. All right, so I think that's really about that. That's just kind of the kind of thing that you're going to need to spend some time with, I think. Now, if you didn't kind of look at someone's pulse rate before, do keep in mind that pulse rate is also going to vary depending on what it is you're doing. Those are folks that stayed in here and relaxed. When you took your pressures after kind of sitting, relaxing, listening to music, you probably saw that not only did your pressures go down, that your pulse rates also went down. Those that walked outside for a little bit, your pulse rates went up. So keep in mind that these things all add together. Just because a pulse rate goes up, yes, that means you have an increased blood flow. So only increasing pulse rate increases blood flow. Only increasing blood pressure increases blood flow. But now you're adding them together. So they have additive effects on blood flow when you both increase your blood pressure and you increase the rate of pulses. Okay, so kind of keep that stuff in mind as well. So now I will basically just kind of leave you all to looking at some of these anatomy things, and if y'all wanted to keep playing with the blood pressure cuffs, you absolutely can. Any questions on anything? Nope. All right, well, let me know. There are also some figures in here for taking pulse points. There's your book has some figures basically for exactly where to find different pulses and things like that too. But once again, they're usually named off of body regions. All right, so y'all get started.